I want to thank the organizers for this invitation to present. I'm going to tell you today about the microbiome and its impact on health and disease. To get started and to put the microbes in context, we are about 30 trillion cells, but at the same time, we are about 38 trillion microbes. Our human cells contribute to 20,000 genes, whereas the microbes contribute to something like 8 million genes and 10,000 species. And I'm going to focus my presentation primarily on the gut. Microbes generally do good. They break down polysaccharides. They help with the synthesis of vitamins and also contribute to pathogens, prevent pathogens from getting in, a term called colonization resistance, and importantly, help mature the immune system. I'm going to tell you today how the microbiome and its changes, a term called dysbiosis, contributes to disease. I've outlined my conflicts here. So what I'm going to start by telling you is how does our gut microbiome develop? How does it respond to perturbations? And how does it change during disease? The microbes are measured by its DNA content, and this could be done by different methods. 16S RNA amplification tells us who's there, whereas whole genome sequencing tells us who's there and what they're doing. So if you're born at birth, your microbiome is acquired primarily from your mother, and you can begin to appreciate that certain microbes, such as the Lactospiraceae and the Ruminococci, are extremely low in abundance at birth, and it picks up over the 3-year period. And bacteria such as bifidobacteria decrease over that time, suggesting that the microbiome is highly dynamic, and this dynamic nature of the microbiome is central to our understanding of how the immune system develops. Now, unfortunately, if you were to prescribe antibiotics, this is one of the early perturbations seen in children or in the newborn. And the earlier you prescribe the antibiotic, the more striking is the change in the microbiota, and you can begin to appreciate the bugs that are mostly abundant, affected by antibiotics are the bifidobacteria. So if you then ask the question, where do these microbes live? You can begin to appreciate that the microbes in the gastrointestinal tract look very different from the bugs in the skin. And if you then inspect this further, you can begin to appreciate that, there are key specific organisms that are location specific. So for example, in the gut, in stool, you see a lot of Bacteroides, whereas in the nares, you see a mix of bacteria. And then similarly in the skin, the microbiome that dominates it are the Propionibacteria and the Staphylococci. So to start off, let's ask the question, how does the microbiome impact the diet, or how does diet impact the microbiome? So very briefly, many factors impact the human microbiome: our lifestyle, our genes, our metabolites, and diet. So what's the evidence that the diet impacts the microbiome? So here's a study that we did in collaboration with investigators at University of Pennsylvania, where individuals were recruited and placed in a clinical research unit and prescribed a defined diet, either exclusive enteral nutrition, which is a fiber-free diet, or otherwise an omnivore vegan diet. And you can begin to appreciate the changes that happen during the dietary phase when bugs are depleted and when they recover. And the take-home messages are: consumption of a fiber-free diet shifts the human microbiome towards metabolism of simple carbohydrates. The lack of dietary fiber slows the recovery after a stress. So if these individuals are exposed to an antibiotic, recovery is slower. Dietary effects on firmicutes alter carbohydrate and amino acid gut metabolites, and this diet-based microbiota metabolites also impact the plasma metabolome. Now, what about another diet? So if you think about a ketogenic diet, another very popular diet, you can again appreciate that the diet, the microbes, and the mucosa, the lining of the gut, impacts how the inflammatory T cells are developed. And a ketogenic diet in the gut microbiome reduces these pathologic Th17 cells, or Th17, or IL-17 producing cell. Similarly, you can extend these types of observations and ask, what about the Mediterranean diet? So what I'm referring to are clear shifts in the microbiome that are defined by the diet. So you can begin to appreciate in a Mediterranean diet to increase these concentration or abundance of bacteria like Faecalibacterium and Prausnitzii or Rosebria. These are bacteria that make lots of short-chain fatty acids. And what studies have shown is that the Mediterranean diet leads to an increase in abundance of taxa associated with low markers of low frailty, improved cognitive function, and a decrease in the abundance of bacteria that are associated with inflammatory disease. And the shift is pretty specific. So if you plot all the bacteria on a whiteboard, you can clearly identify the red dots that are impacted by diet negative in individuals compared to the green dots in those who take a Mediterranean diet. What about diets that we prescribe? Again, you can begin to appreciate here, if you put patients on a gluten-free diet, you decrease the abundance of the Veillonella with a gluten-free diet. Now, there are many diseases, and this list keeps expanding on a daily basis that are associated with the microbiome. And the challenge is, how do you go from an association observation to a causal observation? And I'm going to try to illustrate some key points along the way. So the first disease that I'm going to talk to you about is inflammatory bowel disease, composed of Crohn's and ulcerative colitis. I'm going to illustrate what we've learned about the microbiome by doing a case study. So here's a study where we enrolled 450 children who were treatment naive with newly diagnosed ulcerative colitis. They were treated with the standard of care treatment. So if they had mild disease, they got sulfa drugs. If they had moderate disease, oral steroids. And in severe disease, IV steroids or anti-TNF. And what we learned was that clearly subset of patients with mild disease respond very quickly, whereas patients with severe disease, the response is delayed. And what I'm showing you here is changes in severity over time. So in children who start with severe disease compared to those who start with mild disease, and how the remission in disease change. The key take-home point is that if you were diagnosed with moderate disease, then at the end of 1 year, almost 50% of those children remained with refractory disease. Whereas if you're diagnosed with mild disease, then remission was consistently achieved in about 30 to 40% of children. What were the microbial changes? So as I'm showing you here, plotted from mild, moderate to severe disease, you can see a clear difference in the bifidobacteria over time, and an increase in abundance of oral parainfluenza, Haemophilus parainfluenza bacteria. These are bacteria that normally live in the oral cavity. So clear engraftment of oral bacteria in an inflamed gut contributes to severe disease. What the study also allowed us to do was, by comparing the microbiomes collected at 0, 4, and 12 weeks, we could predict which patient is at risk for a colectomy or is unlikely to require a colectomy. And in this study, 20 of the children required a colectomy by the end of 1 year, and you can see the changes in microbial composition. So what did we learn from this case study? In patients with mild disease to start, 37% of patients achieved remission at week 52. There were significant changes in the microbiota, and clearly this expansion of the oral microbiota. Baseline microbial signature, so collected at 0 and 4 weeks, was predictive of refractory disease. And if you had reduced temporal stability, then you also required a colectomy. And then obviously this question got us closer, but we don't have evidence that says this is causal. So now, while I've so far highlighted my comments focusing only on bacteria, the microbiome extends beyond bacteria. So the microbiome, again, this is about 1 to 2% of our total microbial biomass. And then similarly, virus-like particles. So in this case, 10 to the 8 to 10 to the 9 virus-like particles per gram of stool. What we've observed in patients with Crohn's disease and ulcerative colitis, there is an abundance and change in the microbiome structure. And this is also seen in patients with C. difficile colitis. And very quickly summarizing, the presence or absence of Candida has been correlated with response or no response to FMT, the ability to, and then similarly, this observation in fungi has also extended to viruses impacting disease. The second concept that I'd like to highlight in the context of disease is, does the microbiome explain hygiene hypothesis? So a couple of years ago, we were lucky to study roughly about 1,000 children born in Finland, Petroslav, Russia, and Tartu in Estonia, and followed them monthly for 3 years. And what you can begin to see, if you were born in Finland, your risk of type 1 diabetes was significantly higher. And so we asked the question, by studying the microbiome monthly over 3 years, could we identify microbial findings or features in Finnish children that were different from microbial findings in adult children, in children born in Russia and Estonia? And then could we begin to find associations with the risk of type 1 diabetes? So what we identified is that by looking at the microbial complexity in children who were at risk of type 1 diabetes, so seroconverted to type 1 diabetic antibodies, they shifted their microbiome structure 1 year before they got type 1 diabetes. So a marker that tells us about disease susceptibility. And then when we looked at the microbiomes in children born in Finland compared to those in Russia, in the first year of life, you could find that the Bacteroidetes are more common in Finland and Estonia, and the actinobacteria are highly abundant in Russia. So what Tommy Matanen then asked is, does this correlate with function? So when he looked at the functional pathways that are affected by these bacteria, the dominant pathway was the pathway that tells us something about lipid biosynthesis or lipopolysaccharide biosynthetic processes. And so what Tommy and collaborators did was to isolate these individual bacteria. So if you were born in Russia, you had a E. coli dominant LPS, whereas if you're born in Finland, you made many different flavors of LPS. And when we then purified these different LPSs and tested whether they can increase the risk or protect you from type 1 diabetes, and I'm showing you one experiment where we took NOD mice, as these are mice that are predisposed to type 1 diabetes. And if you gave them the 6 acyl chain LPS, the LPS made by E. coli, their onset of disease was delayed. Whereas if you gave them the 5 chain acyl LPS, which was common or more dominant in the Finnish kids, you ended up with diabetes coming on a lot earlier. So what we learned out of this is that, there is an important component of immune education. And if you were born in Russia, you were exposed to LPS, E. coli LPS, and that stimulated the mucosal immune system and generated an educated immune system that decreased the risk of autoimmune disease. Whereas in the case of Finland, you saw a 5 acyl chain LPS that resulted in a non-educated immune system that increased the risk of autoimmune disease. So the question is, are we losing our friends? Now, obviously, it's in any talk one has to refer to the current crisis with COVID. And so here a number of investigators have looked at the microbiome in small subsets of patients with COVID-19 disease. And here's a study that looked at the fecal microbiome collected in 15 patients with COVID. And you can begin to appreciate that symbiotic bacteria, or the good bacteria, were markedly depleted in individuals infected with COVID, allowing for the expansion of opportunistic pathogens that correlated with severity of COVID-19. So what we identified here is that the composition of the microbiota in patients with COVID-19 is concordant with disease severity and the magnitude of plasma concentrations of several inflammatory cytokines, chemokines. And in patients with COVID-19, we found a depletion of bacteria that make those short-chain fatty acids. So, for example, Faecalibacterium prausnitzii or Eubacterium rectale, and several of those bifidobacteria species that I initially told you that these are important colonizers in the newborn. And in the short-term follow-up studies, this altered microbiota appears to persist even after clearance of COVID-19. Now, obviously, in addition to the inflammatory syndrome, does COVID-19, it does ACE2 expression, the core receptor for COVID-19, also have an impact on anti-cytokine therapy. And what you can begin to see in a study from 4 different institutions, you can clearly begin to appreciate that the expression of ACE2 changed in the small bowel and the colon. And by treating these patients with anti-TNF drugs, we were able to return or bring back the ACE2 levels to non-IBD status. So impacts the disease, alters the microbiome, and in patients with IBD and alterations in ACE2 expression, the ACE2 expression could be corrected by anti-cytokine therapy. What about bugs and drugs? And so to start off, proton pump inhibitors are commonly prescribed drugs, and you can begin to appreciate with increased use of PPIs, you alter the commensal microbiota. So here focusing on 2 classes, the gamma protobacteria and the lactobacilli, you can begin to see the increase in abundance, whereas the actinos and the bifidos decrease in abundance with PPI. Now, in addition to the effect of PPI, the microbiome also alters the bioavailability of certain drugs. And here to illustrate a few examples, so sulfasalazine, we've known, needs to get cleaved by azoreductases that are bacterially derived into the active moiety 5-ASA and sulfapyridine. And then similarly, methotrexate is also broken up into the active moiety and an inactive metabolite. And this extends also to antibiotics. So bugs impact the diet, bugs contribute to disease, and also alter the drugs that are used to treat disease. What about microbiome and cancer? And so in the recent years, we've begun to appreciate the role of the microbiome in all types of cancers. And in an elegant study that was published last year, by examining 7 cancer types across 1,500 samples from 9 medical centers, so broadly distributed, the findings that certain tumors have a richer and a diverse microbiome, and that there are microbiome signatures that are different in different tumors was very clearly highlighted. So in this plot that I'm showing you, if you look at the various cancers going from melanoma to breast cancer, you can begin to appreciate how the Shannon diversity index changes over time. And then if you go and dissect the microbiota inside cancer, you can begin to appreciate that these microbiota invade the cancer microenvironment and bring about immune responses that negatively impact responses to treatment. And you can dissect this even further. You can begin to appreciate there are different species level phylotypes across different tumors. You can appreciate significant differences. For example, if you look at breast cancer compared to pancreatic cancer. And then importantly, the microbiome also changes if you are ER positive versus an ER negative breast tumor, or a HER2 positive versus a HER2 negative breast tumor, suggesting that prevalence of multiple taxa also different between a cancer type, in this case, breast cancer. What this study also further identified were that there are bacterial pathways and taxa that were different in patients with lung cancer who were smokers or compared to never smokers. I've already told you about ER positive and ER negative breast cancer. And then bacterial taxa also contribute to whether a melanoma responds to checkpoint inhibitor therapy or incompletely responds to therapy. So a broad impact of the microbiome on cancer. And what I thought I'll do towards the end is to sort of begin to tell you about what are some of the potential levels of impact for the microbiome in treatment. So the likelihood is that as we move from associations of the microbiome with disease, I've told you so far that there are clear signals that contribute to inflammatory bowel disease. I've also told you that the microbiome at birth impacts development of the immune system, and there I've highlighted a few examples of how the microbiome contributes early in life with certain taxa that expand with time, and early exposure to antibiotics ends up depleting those good bugs, the bifidobacteria. And then I've gone on to illustrate the concept of the hygiene hypothesis on how the microbiome might impact the susceptibility to type 1 diabetes. And in the last example, I told you that the microbiome across cancer types are different, and that within cancers, if you take breast cancers or lung cancers, they have a clear microbial signature differences, whether you are ER positive or negative, or a smoker or a non-smoker. And in the remaining few minutes, I'll begin to talk to you a little bit about how the microbiome-based therapies might impact disease biology or help us return to health. So the options are bugs as drugs. That's what we do now in the form of FMT. You can identify chemistry or molecules made by bugs, and you could give that in the form of a pill. The molecules made by bugs are made by a collection of genes, and we refer to this as biosynthetic clusters. So one possibility is to express these biosynthetic clusters in a probiotic and then give it to individuals so that when the probiotic gets to the terminal ileum or the colon, the biosynthetic cluster is activated, results in the production of a molecule that can impact disease. And then finally, you could also use bugs to modulate disease. And I'm going to give you a few examples here. So the first example is Clostridia. So we've been doing this now for a long time, and in an elegant study published now almost 8 years ago in the New England Journal of Medicine, fecal microbiome transplant therapy was used to treat patients with Clostridia. So what I'm showing you here is that patients with infection with Clostridium difficile undergo a marked shift in the microbiome. So you can begin to appreciate that there is a huge expansion of the Bacteroidetes and the Firmicutes, whereas the other bacteria change in abundances. And obviously, metronidazole and flagyl treats a subset of these patients, but the efficacy of antibiotics and the first recurrence is significantly high. So what this initial study did was to pre-treat the patients. So the idea was to give antibiotics to start with, and then do a fecal microbiome transplant. So creating space for the introduced bacteria to get in, and to ask the question, does the C. difficile colitis get better? So what you can see here is that clearly the percentage of patients cured without relapse was significantly higher in patients who got infusion of donor species compared to the individuals who got vancomycin or vancomycin with bacterial lavage. And what was more striking is that in individuals who got the microbiome from a donor, their microbiome shifted to look more like a donor, whereas the microbiome in patients with C. difficile colitis pre-treatment was markedly depleted. Example that has got us closer to the causality discussion is a condition called environmental enteric dysfunction. So this is a common gastrointestinal disturbance in children born in the developing world in the setting of inadequate nutrition and recurrent infections during the first year of life. And ED, when it was first described, was seen in Europeans who also traveled to the tropics and again, through a sequence of events, ended up with endoscopic and histologic appearance that showed us that the microvilli were markedly shortened, and you ended up with flat villi. What these studies, most of them led by Jeff Gordon, have taught us is that it's triggered by a diet that is low in protein or fat, results in increased colonizations of pathogens that are hard to clear in these children, and then the immune cells also function poorly, and the microbiome diversity is altered as you get environmental enteropathies. What Jeff Gordon and colleagues did were to go on to identify, by studying children born in Bangladesh who developed ED and those in Malawi, he was able to identify a community of 15 bacterial taxa that, when present, contribute to normal development of the gut microbiome during the first years of life, 2 years of life. And the experiment that Jeff did was that changes in abundance of these taxa provide a way of defining severity of microbiota perturbations in these malnourished children. And by characterizing the microbiome and then replacing the microbiome, as shown in this plot here, if you look at days to start of inflammation compared to weight to length z-scores, you can begin to appreciate that children who got the microbiome with who got microbiota directed complementary food formulations recovered in their height to weight ratios compared to those with the ready to use supplementary food. And this was seen in over 100 Bangladeshi children. So clear benefit of the MDCF2 over the ROSF. So I've given you a few examples of, in the first example, I told you about FMT and treatment of C. difficile colitis. And in the second example, I've used environmental enteropathy to illustrate the principles of selective microbiome replacement. Here we have, in a series of studies done by Jeff Gordon and colleagues, they identified the depletion or the absence of these 15 microbia in the context of a poor diet leads to environmental enteropathy. And if one were able to replace that 15 species cocktail, one was able to correct features like height to weight and growth over the following 2 years. What about microbiota as a predictor of mortality in allogenic hematopoietic stem cells transplantation? So again, an elegant study that Eric Pema and others have built out. The gut microbiome diversity declined during the course of allogenic hematopoietic stem cell transplantation. And this was seen in 4 different institutions, all the way from Memorial Sloan to Hokkaido in Japan. Higher the diversity, better the patients did. And clearly, overall survival was longer among patients with high diversity, both at Memorial Sloan Kettering and the other 3 institutions, suggesting the microbiome diversity helps with response and successful HCT. What this study also went on to show is that high diversity among patients at Memorial Sloan was associated with a lower risk of transplant-related complications. And this is both plotted here, for example, showing that higher diversity is associated with lower transplantation mortality, and then overall survival was also seen as a feature with individuals who had higher diversity at the time of transplantation. So what might be the future ahead? I've given you the principles of understanding the microbiome and how they contribute to disease. And so I think what these types of studies have begun to highlight is that it's time for us to sort of reconsider how one might define probiotics. So based on the examples that I've given you, I've highlighted how we might rethink probiotics. So I told you in a preterm neonate, the bifidos in its abundance are markedly depleted. And so maybe the probiotic there needs to be enriched for bifidobacteria. Similarly, as you grow towards adulthood, you have the risk of inflammatory diseases. And so if you can then protect, these not so friendly bacteria coming and sticking to the lining of the mucosa. So if you can identify probiotics that help with adhesion properties, then one could think about potential treatment. In conditions like IBS, inflammatory bowel disease, certain mood disorders, you can now begin to appreciate that changing that, dialing up and dialing down that microbial mixture might provide us clues towards disease resolution. And looking forward, one could begin to think that there could be microbial signatures that tell us about Crohn's disease natural history, predictors of those who need surgery versus those who need a change in biologic or stopping a biologic. I've told you about how fecal microbiota transplantation can correct certain defined defects in the human genome. I told you about how the microbiome changes in different cancers. One could begin to come up with a screening test that might tell you something about colorectal neoplasia. I didn't spend a lot of time talking about cancer immunotherapy, but there again, recent studies have shown that the microbiome community structure defines a responder versus a non-responder. And one could, get to a stage where one takes into account the features of the individual, their diet, their exercise activity, their type of work, and then come up with personalized predictions of what the replacement microbiome needs to look like. And one could potentially imagine this type of interventions, from prediction to the right treatment, to screening, to treatment responses that could have a microbiome cocktail that's associated with that. So I think I'll stop there. And what I've tried to do is given you sort of a broad overview of how the microbiome impacts health. I've highlighted a subset of diseases going from inflammatory disease to cancer. And then I've told you about how the microbiome impacts drug bioavailability, its diagnostics in the context of disease, and then alluded to you about opportunities for treatment. I think I'll stop there and I'll be happy to take questions.